So good afternoon, everyone, and thank you for joining the Young Investigator seminars. Today we have Barbora Smolková, who is going to talk about mitochondria, liver development, and diseases. Barbora is already a postdoc, right, in the physics center in Prague, and she's with in the group of Oleg Lunov, and the director of the department is Alexander Denieska, right? So maybe well, you can tell us a little bit, Oleg, about Barbora and the stay that she did with Gareth Sullivan in Oslo and what she accomplished.
Yeah, so I will explain a bit, brief introduction. So Barbora did a PhD at my group, very good, very excellent work, and then went for mobility to Gareth Sullivan. It just actually a very great opportunity to for the cost, and thanks for the cost to have this network that we could build, that we find out about each other, that we can do some really interesting things together. Actually, we never met with Gareth personally, only via internet, but somehow the collaboration works very perfectly. So she went there to learn techniques on organoids, and she will show some very nice data what she got already. And she will show that it's very challenging to work with organoids. You need at least 21 days to get the thing done, at least to grow it up, and it not always works as you want to. So within even this small time of half a year that she had a mobility, she gained really good results and really propelled our research and also interaction between two groups. But not only those two groups, I'm really thankful for the cost action that we could meet each other and create a great teams. Okay.
Oh, that's great. That's great, Oleg. You are talking about Gareth
Yeah.
Sullivan.
Yes.
Oh, that's fantastic. That's the spirit and the main objective of this cost action in general. I'm very pleased to know that. Very good. We are really looking forward to hear what Barbora has to tell us.
So Barbora, you can start when Barbora, sorry, you can start whenever you want.
Okay, thank you for introduction. I will just share my screen. Just 1 second. So I hope you can see my presentation now. Yes?
Perfect.
Okay, so thank you very nice for Oleg's kind introduction and for invitation of Javier for this opportunity to have a talk here. So I will start with so I will start now with my presentation. So I hope you can see the switching the slides. Yes. Okay, great. Thank you. Then it's working. So during my PhD, I focused on redox system modulation in liver cancer cells, and I analyzed 2 different approaches of physics-based therapies. One is non-thermal plasma, the other one is laser irradiation. And both of those methods, they showed great potential in biomedicine, but both are lacking some very precise mechanistic understanding on how they interact with living cells. So we, by the study, a little bit contributed to this understanding. So just very briefly, I will explain what I did. Non-thermal plasma is ionized gas, and it's containing of different reactive oxygen and nitrogen species, UV photons, and other uncharged particles. And we analyzed the effects of this non-thermal plasma in 3 hepatic cancer cell lines, and we showed that the Alexander and HUH7 cells, they were less resistant to plasma in comparison to HepG2. Those cell lines are closely related, but they showed very different genetic background and morphology. Indeed, HepG2, they are having elevated level of BCL2 proteins, which is well-known anti-apoptotic proteins. So when we block these anti-apoptotic proteins, we could see that the cells were sensitive to plasma treatment. Moreover, we showed in our Western blots that non-thermal plasma led to elevated level of BCL2 in HepG2, and the cells were protected in this way from plasma treatment. On the other hand, HUH7 cells, they are having elevated level of p53 protein, but the mutated form, which is mostly connected to higher resistant to treatment and very aggressive tumorigenicity. And we are showing that plasma led to downregulation of this mutated form of p53 here. So in this way, we explain mechanistically the effect of non-thermal plasma in liver cancer cells, and we are showing that in one case, the plasma led to mitochondrial damage and p53 modulated apoptosis, and in other case, the plasma due to elevated level of HepG2 of BCL2 in HepG2, this elevated level contributed to the resistance. In the second part of my thesis, I focused on laser irradiation, and we analyzed 3 different laser wavelengths on 3 different cancer cell lines, which I mentioned before, and I will just briefly explain what we did. And we show that the cytochrome C oxidase, the complex 4 of ETC, is the major sensor and effector of the laser light irradiation in cells. We hypothesized that the blue laser irradiation inhibited the COX function due to depolarization of the mitochondrial membrane potential, led to the increase of ROS, depletion of the ATP, and subsequent cell death, whereas the red laser irradiation activated the COX function, and due to hyperpolarization of the mitochondrial membrane potential and increased ROS, also led to cell death. We validated our hypothesis by different activators and inhibitors of complex 4. Here I am just showing the results. Sorry. Here I'm just showing the results with the specific inhibitor of cytochrome C oxidase, which is called ADA5, and we could see that this co-treatment of ADA5 with red laser irradiation were able to decrease the ROS, stabilize mitochondrial membrane potential, and even to protect the cells from cell death. But this was not the case with blue laser irradiation, where we used the specific activator, and then we could see the stabilization and protection of cells from cell death. On the other hand, we also noticed similar response of HepG2 cells to laser irradiation, and we thought that this is again due to BCL2 protein. So when we used for HepG2 the inhibitors, which we used previously with plasma, we again saw a cell death. But we wanted to confirm the BCL2 involvement even more, so we overexpressed BCL2 tagged with GFP in Alexander and HUH7 cells, and then we could notice that these cells which were overexpressing BCL2 were again protected from cell death. induced by laser irradiation. In addition, due to our fancy confocal microscope, we could see that the BCL2 was translocating to the mitochondria upon laser irradiation, and it has been shown that this is the way how BCL2 may counteract apoptosis. So after finishing my PhD work, just after graduation, I moved from Prague to Oslo, and where I joined the group of Gareth Sullivan, and I met a lot of great people at Department of Pediatric Research at University Hospital of Oslo. And because Norway is very famous for work-life balance, I didn't spend all my time at lab, and I also had the time to explore beautiful nature of Norway. So now I would like to tell you more about what I've done in Norway. So liver is the primary site of drug metabolism, and hepatotoxicity is the leading cause of drug development failure. Now it's clear that we need that both these toxicity studies and drug development needs better in vitro models for those studies. Especially, there is need for models which better representing the complexity and very complicated liver architecture, and which are better mimicking the conditions in vivo and high level of organization liver. Because liver doesn't contain only hepatocytes, but I don't think that I need to explain this more here. So current models, including mostly the cancer cell lines like HepG2 or HepaRGs, but they are possessing, they are showing many limitations and do not recapitulate the liver function so much as it is in vivo. So another models which are used for hepatotoxicity studies are primary human hepatocytes, which are great, but when they are longer in culture, they are losing their function. There has been few 3D models which are better mimicking the in vivo conditions, but some of them are again showing limitation. Some of them are containing just one cell type or even more cell types. There has been shown recently the hepatobiliary organoids or different kind of spheres, which again, each of them is having also still some limitation. So there is really need for better and reliable in vitro models in hepatotoxicity studies. Recently, induced pluripotent stem cells have gained attention in this area, mostly because of their unique properties of self-renewal and pluripotency. And this enables the differentiation of the cells of induced pluripotent stem cells, patient-derived, into different kind of cells. And when this is combined, the usage of induced pluripotent stem cells has shown great potential to mimic in vivo condition, also when used for 3D organoids generation. And this can be very useful models for drug screening and liver diseases modeling, and maybe in future for regenerative medicine and personalized medicine. Majority of the protocols for stem cells differentiation into hepatic cells includes and following the liver development during the embryogenesis. In vitro condition, in vitro conditions, there has been additive of different growth factors, which are then contributing to the development of the liver. Recently, this addition of growth factors has been replaced by small molecules, which are dramatically reducing the cost of this kind of generated either organoids or hepatocytes. So in our work, we used the organoids which were produced by Garrett's group. They replaced the growth factors by small molecules, and they are showing that the whole process is starting by the formation of the aggregates, and these aggregates are still maintaining the pluripotency. And then by addition of different small molecules at specific time points, the differentiation is following the liver development in vivo. Single cell RNA sequencing has shown that the liver organoid produced by Garrett's group, they are representing the liver, and also they compare their liver organoids by the protocol developed by another group, and they are showing different population of different kind of cells, not only hepatocytes but also Kupffer cells, cholangiocytes, endothelial cells. Moreover, they are showing the organization of liver organoids. On the top of liver organoids, they are mostly epithelial cells, and these are hepatocytes, and then under there is a population of stellate cells, which is shown also here in this cross section. Moreover, they are showing different population of endothelial cells, and if you are more interested in those organoids, there has been published preprint with all the characterization, which is really cool. Moreover, they are showing that the produced organoids are functional and that they are metabolically active, that the cytochromes are active and the metabolism is working. They show the bilirubin uptake and urea secretion and much more, again in the preprint. So I focused mostly on mitochondria activity in those liver organoids during the first days. So mitochondria are double membrane structures, and they are involved in many functions of the cells. They are involved in energy production, through oxidative phosphorylation, in amino acids and fatty acid metabolism, in production of reactive oxygen species, in calcium homeostasis, but also in apoptosis. Mitochondria are very dynamic structures. They can fuse or divide under specific condition, either physiological or stressed. Recently, it has been shown that mitochondria are also very much involved in the process of differentiation and development. It has been shown that pluripotent stem cells and stem cells in general, they are having high levels of glycolysis. They maintain high levels of glycolysis. Moreover, it has been shown that the mitochondria of stem cells are mostly globular, with poorly developed cristae and with very low production of reactive oxygen species. However, when cells are being differentiated during the first hours, there has been metabolic switch from glycolysis towards oxidative phosphorylation. So in differentiated cells, the mitochondrial network are more developed. Mitochondria are more tubular with well-developed cristae, and those cells prefer oxidative phosphorylation. The crosstalk in between mitochondrial metabolism, pluripotency, and differentiation has been shown already, and this is very complicated thing, and many metabolic pathways are involved in the regulation. And deep understanding of this crosstalk between mitochondrial remodeling, metabolic switch is very crucial for improving our knowledge about the mechanisms involved in lineage-directed differentiation and tissue maturation. In addition, this understanding is very important for developing of better models, even for mitochondrial diseases. So we could use patient-derived fibroblast containing mitochondrial mutation, reprogram them, and then this can help us for better understanding of the disease, or even to more personalized treatment, better understanding of the disease origin. Here I would like to show you one example of Garrett's previous work and his colleagues. They used cells which possess polymerase gamma mutation. Polymerase gamma is a polymerase which is involved in the replication of mitochondrial DNA. And so they have a look on mitochondrial remodeling during differentiation into neurons, and they can you hear me? And previously?
I can hear you now.
Because I just got a message from Oleg that he can't hear me. Can you hear me?
Does everyone hear, or I have problems
fine.
with the?
I can
connection?
hear very
Yes,
nice.
yeah.
Yeah,
That's
me
fine.
too. But there was no break in the sound.
Okay, so
Hmm.
okay, so no problems with hearing.
No.
Okay, so thank you, and I can continue then. Okay, so they used the stem cells normal and then derived from the patient with polymerase gamma mutation, and they show that this mutation led to the abnormal mitochondrial remodeling in the neural development, and they are showing that this was due to the downregulation of this pathway, which is very much involved in mitochondrial biogenesis. So this was just one example how the better understanding of the mitochondria during the development and during the differentiation may help to create better models. But there are still a lot of questions which we don't know the answers for. We just know that this metabolic switching during the differentiation is germ layer specific, but there is really lack of extensive characterization of the mitochondria in different stem cells differentiation models. Moreover, we don't know much about the changes in if there are some changes during the differentiation in the compartments of mitochondria, like the membrane, proteins, or mitochondrial DNA, and the changes in mitochondrial activity during the mitochondrial biogenesis process. Moreover, the kinetic of mitochondrial biogenesis during differentiation is not well described, and we don't know which signaling pathways are determining those changes and how mitochondrial network is remodeled during differentiation. Also, the question is if those changes in the structure and function of the mitochondria, which we could see in the pluripotent state and during the differentiation, are just the cause or the consequence of all those processes. And last but not least, we don't know if maybe mitophagy is very important during this process. So during my staying in Oslo, we tried to answer or try to analyze some of those questions. And so we mostly focused on hepatocytes, and this is study which was done previously, and because I couldn't find many studies which focused on mitochondrial analysis during hepatocytes differentiation, and this is one which I found where they follow the changes during the differentiation, and they are showing that the mitochondria in differentiated cells were more tubular-like and in comparison to undifferentiated, and they even are showing the increased content of mitochondrial DNA and increased activity of enzymes of oxidative phosphorylation. However, the bone marrow mesenchymal stem cells are not very appropriate model to study the hepatocytes differentiation. So in our work, we use the organoids which I described previously, and we focused mostly on the first 7 days in here during the differentiation. So I learned this technique of organoids production in Garrett's lab, and I hope successfully. So we isolated the protein and DNA content of the organoids at each day. And at day 7, we checked if the differentiation was successful by staining for HNF4 alpha, which is a hepatocyte marker. So from isolated proteins, I'm just showing very preliminary data. Now, we observed the increase of cytochrome C oxidase, which is the complex 4 of electron transport change during this process of differentiation, with subsequent decrease of TOM20, which is mitochondrial outer membrane protein. This may suggest that the mitochondrial activity is increasing very dramatically during the process of differentiation. However, the content is decreasing. We also analyzed the content of mitochondrial DNA. TFAM is the protein which is directly proportional to content of mitochondrial DNA, but we need to validate those changes by PCR. Moreover, we realized that the 2 mostly used housekeeping genes, actin and GAPDH, are not serving as a proper control, because during the process of differentiation, everything is changing. So in order to avoid any technical issues during our experiments, we did the total protein stain post blotting. Further, we want to determine the ratio in between mitochondrial and nuclear DNA. So we use 2 approaches for DNA isolation, one kit based on the column and proteinase K cleavage, and the other one was based on the magnetic beads, and we used the automated machine for isolation, but we noticed that we have very low ratio of mitochondrial to nuclear DNA, in between all up to 4, and we expected like hundreds to thousands written numbers, so we have some problems. And this is one of the troubleshooting of the Borg with organoids. So we are losing either the mitochondria or they are being fermented during the extraction. Further, we would like to analyze the mitochondrial dynamics and their activity by live cell imaging. And live cell imaging is bringing many problems. One of them is that we can introduce a photo bleaching or other artifacts by imaging. So selection of appropriate imaging method is very important. And thanks to the we have really great system back in Prague for imaging our spinning disk confocal microscope, but unfortunately we need it also in Oslo. So, and thanks to the cooperation with Professor Anna Simonsen from Oslo University, we got access to dragonfly spinning disk microscope, and we established, I think, quite nicely the staining of the mitochondria in liver organoids. Here you can see the JC1 staining, which is showing the level of mitochondrial membrane potential in liver organoids. So here are just reconstruction of 2 pictures from different days of organoids growth. So further, we would like to follow those changes in mitochondrial dynamics and in mitochondrial membrane potential during first 7 days of organoid differentiation. Further, we were interested if mitophagy is involved in the process of differentiation. Cell is in order to keep the homeostasis of mitochondria in cells, they have different, they are using different levels of mitochondrial quality control, and one of them is mitophagy or mitochondria specific degradation by autophagy. Previously, Garrett's lab, and they showed that autophagy is very important during the differentiation and may modulate cell fate decision during the lineage commitment. In their study, they showed that degradation of SOX2, which is mediated by autophagy, may facilitate the differentiation of cells toward mesoendoderm. And but when they downregulated the autophagy, and this led to the maintenance of SOX2, this promoted the differentiation of cells toward neuroectoderm. And we again isolated the proteins during the first 7 days, and we observed some changes in LC3 and P62 during the process of differentiation. Both proteins are involved in autophagy, but for now we can just speculate what is the role of them in here, and we would like to unfold this more by using very fancy cells, which were constructed and published in the autophagy paper, which I mentioned before. those induced pluripotent stem cells, they are having double-tagged LC3, which is inducible by doxycycline. And double-tagged, it's because it has two proteins. One is mCherry, the other one is GFP. And once the protein is in lysosomes due to the sensitivity of GFP to very low pH, we could see just the red puncta. So the GFP is quenched in the acidic environment. So we would like to now use this double tag cell line to follow the changes during the first few days of differentiation. But in addition, we have another double tag cell lines. The first one is which are working in the same way as LC3, but the first one we would like to follow the mitophagy, because this one is having the double tag MLS. MLS, it means mitochondrial localization signal. And the other one are having the double tagged P62, another important protein involved in autophagy. So in next few weeks, we would like to follow the changes by using those cell lines. So there is still a lot of work ahead. We would like to optimize the mitochondrial and nuclear DNA isolation. We also want to focus on mitochondrial membrane potential and dynamic analysis during the first 7 days by using live cell imaging, by using the dyes which we established the labeling with. Moreover, we want to focus on ROS signaling during the process of differentiation and to analyze the signaling pathways and the molecular mechanisms which is behind this. So the next step for our work is the differentiation by using the reporter cell lines, and in future maybe to use the differentiation and liver organoid production by using the cell lines which are from patients with polymerase gamma mutation, and to analyze the mitochondrial changes in there. So with this, I would like to thank to Cost for hosting me here. I would like to thank to Gareth for hosting me in Oslo. To Sean, this is Gareth postdoc. With him, I did most of the work. To Safira and to Maria from Department of Research of Institute of Internal Medicine. Moreover, to Professor Anna Simonsen from University of Oslo, and of course to Oleg for supervising me during my PhD and for this opportunity to send me here. So thank you. And if you have any question, I would be very happy to answer them.
Okay. If
Great
someone
job.
has questions, please ask. Javier asked me to handle the Q&A session. There is one question in the chat. Have you done or are you planning to do organoids with knockout genes?
I haven't done that, but maybe Gareth can elaborate this more, because in his group, some people are working also in this direction.
Yeah, well, it's not really knockouts per se, but what we're playing with is we're using patient-specific iPSCs to make organoids, and then we're doing gene correction to make isogenic control. So it's equivalent to sort of you have the mutated form of the protein, and then you correct the protein to its wild type form. So we do a direct comparison. We are in the previous study, we used an ATG7 knockout line that we generated. So we have done those as well.
Barbara, this is Carlos Lopez from Maribel Lucena Lab.
No,
It was
short lab.
okay, the same. First of all, congratulations for your talk. It was a very nice work. I have, it's not a question, actually, it's a suggestion. Can you go back to the presentation, to that slide where you show the levels of COX-1 and TOM20?
Just 1 second, please.
Because you say something about the activity of COX-1, but that's a Western blot, right?
Yes.
So what you are measuring there are steady state levels. So if you want to measure activity, you have to do a biochemical assay.
No, I didn't mean the activity.
Yeah.
I mean that we could observe some changes in the expression. Yeah, okay, that was my mistake.
But the way I interpreted this data is that you have higher mitochondrial mass because you have a more complex one.
Lower.
But then what you have decreased membrane.
No, I think I don't know precisely because we will need to have the data from microscopy. But we think that we have lower mitochondrial mass because of the TOM20, but they are more active because oxidase level is increasing. But of course, we would need to do other studies to, as I told, that these are very preliminary data, and we need to elaborate it more, of course.
Yes, I think you have to take picture, but I think what you have is enlarged mitochondria, like longer mitochondria instead of rounded ones, which is what you might expect if you are differentiating the cells.
Yes.
Okay.
Thank you.
Thanks for the question. Someone else? Maribel, do you have something?
Well, I am really impressed. Very, very nice work. That's very difficult, very demanding, and very and the technology also that you can offer with microscope is fantastic. For me, when I heard about the POG mutations, what I could link that to the valproic acid hepatotoxicity. But you have talked about those mutations during the regulation of also neuronal differentiation, no? So I don't know if that has could be seen also for in the liver.
Yes, in fact, the polymerase gamma mutation are associated not only with neural defects, but also with liver.
In liver, no?
Yes, with liver failure. Yes.
Yeah. There is a link though between the brain and in a sense. It's because the patients present with epilepsy, and that's what's normally that kills the patients, but then they're prescribed valproic acid and then they suffer liver failure due to valproate toxicity.
Yes. So
it's quite interesting. That's what drew us first into looking at this. But in the most severe Pol gamma cases, Alpers-Hutschlocker syndrome, which is very rare, they have spontaneous liver failure as well, these patients, and prognosis is pretty poor. It's like between 18 months and 7 years old. The patient normally is dead, so it's not a very nice disease to have. Fortunately, it's very rare.
Yeah, that's very interesting. And now you have moved to pediatric uni, haven't you? Yes. I'm working on pediatric patients. Oh, well, your lab is at the pediatric unit, but you are doing the same that you were doing before.
Yeah, we're very fortunate because, as Barbara said, it's a really nice environment, and there's a lot of interaction between clinicians who are, very translationally orientated, and so we have access to a lot of different patient material for metabolic diseases. And for example, we're looking at one called Augen syndrome, which is a Norwegian disease. There's about 100 cases worldwide. So it's a episodic cholestatic disease, which basically manifests at puberty. So we've got to the bottom of that as well, but that's through purely being based in the pediatrics department, which is, it's very nice in the sense that we have access, and the patients and their families are very supportive on being involved in studies as well. So it's a fantastic environment to be in.
Fantastic. That's very fortunate indeed. Yes. Congratulations, Gareth.
Yeah.
I'm very happy to learn that this collaboration has been so fruitful. And
I'd like to say as well that Barbara's been brilliant. So it's been, we've been very fortunate. Obviously we have from Javier's lab and now from Oleg's lab, the quality of of the researchers has been very good and very and she's fitted in perfectly. She's been a joy to work with.
Great.
Thank you. I also very much enjoying the time in Oslo. Yeah, I met the wonderful people, learning this technique of work with induced pluripotent stem cells and organoids production, because previously I just worked with hepatic cancer cell lines. So this is really amazing for me. So thank you.
Yeah. The figures were fantastic to see the organoid and the lining of the hepatocytes. It's really fantastic. Amazing. There is no more question.
Yes, very great that we got in touch with Gareth to have some more elaborate models of the liver. That's very nice.